want to spend today talking about the involvement of viruses in cancer. And in the end, I want you to remember, if you only remember one thing, viruses themselves don't cause cancer. They lead up to cancer. They cause changes in cells that predispose them to cancer. So we're going to talk about transformation in oncogenesis. Oncogenesis is the induction of cancer. Transformation is something different that leads to it. So let's first define what transformation means. Here's an experiment. On the upper left, we start with a hamster embryo. Chop it up and digest it with trypsin to make cells out of it. And we plate cells on a dish, a culture dish, and grow them. These cells will grow for a while, as you can see in the graph on the right. But eventually, after 20 or 30 doublings, they'll die. And this is typical of most cells, what we call primary cells that you make from a tissue. If you took some cheek cells from you could plate them out, they would grow 20 to 30 times, and then they would die. They are mortal. They don't live forever. However, in this experiment, in some cells, if you just let them grow, a few will survive, and then they will grow and become immortal. Or you can speed up the process by treating these cells with chemicals, mutagenic chemicals, or irradiating them, in which case you will cause them to become transformed and become immortal and live forever. So on the little experiment on the left where it took our hamster and plated out cells, we have one plate we haven't treated. These will grow only for a certain time unless we transform the cells in some way. So transformation is changing the ability of cells to grow so that they're now immortal, and they have a number of other properties associated with them as well. The first is that if you look at them under a microscope, they look very different. On the left at the bottom here is a normal sheet of cells. These could be primary cells that you've just plated out from a tissue. They look good, they're flat, they touch each other, they stop growing. On the right is a transformed culture of cells. They tend to keep growing. They don't respect each other's borders. They pile up on one another, and of course they grow forever. So that's just a morphological difference between normal cells and transformed cells. This period in which they die and new cells reemerge is called crisis here in this experiment. Now, transformed cells have been known for many years in laboratories, and they have a number of interesting properties. We've already looked at the morphology of the cells, and if we look at the side of these cultures, you can see the normal cells, when they touch each other, they stop growing. The transformed cells keep growing, they keep dividing, they pile up and make stacks of cells and so forth. So a number of properties of these cells include they're immortal. They grow indefinitely, like HeLa cells. These were the first immortal cell line isolated from a human, and they were found to grow forever. Turns out they were from a tumor that was already immortal. That tumor would live forever had it not killed the host, Henrietta Lacks. Loss of anchorage dependence. The cells, when they touch each other, they don't stop growing, but you could also grow transformed cells in agar. You could pour a thick layer of agar into a plate and mix the cells in with it so the cells are suspended and they'll grow. Normal cells won't grow suspended in space. They like to be anchored to a flat surface. Loss of contact inhibition we've talked about. Colony formation in semi-solid media, I've just told you. And they have decreased requirements for growth factors. They need less serum than primary cells in order to grow. The reason for this will become apparent in maybe 10 or 15 slides. So that's the property of transformed cells. So that's transformation. We're going to talk about how viruses transform cells, but already you should know that you can take a normal cell and transform it by mutagenizing it in some way. The other half of today's lecture title, Oncogenesis, is the development of cancer. And cancer, of course, is a tumor which swells caused by abnormal growth of tissue. Tumors can be benign, where they're self-contained and the cells don't migrate elsewhere in your body, or they can be malignant. They can metastasize and make small tumors elsewhere, which will eventually, of course, kill you. Cancer is a genetic disease. Even the cancers that you'll see induced by viruses today. It's a genetic disease in the end. There are about 8 million deaths a year from cancer in developed countries. You need about a dozen mutations in your genome that affect signaling pathways, which we'll talk about today, involved in cell proliferation, survival, determination of cell fate, and maintenance of genome integrity. About a dozen mutations, and boom, the cell is going to be cancerous. So this is all about altering the regulatory pathways in us that ensure that most of our cells don't divide all the time. As I said to you earlier when we were talking about viral DNA synthesis, most cells do not divide. Many viruses make them divide because they prefer that. They need it to have the DNA polymerase and so forth available to them. And the pathways that regulate cell division, as well as survival, cell fate, genome integrity, those are the genes that mutate in cancers. And that's why now, whenever someone has a cancer at a major medical center, an academic medical center, we want to sequence it. And we put the sequences into a database so we can start to accumulate banks of information about what kinds of genes are mutated in human cancers. And we can see if people are born with 6 of these, then you're predisposed to some sort of cancer, and we could maybe do something at some point in the future. So you can inherit some of these mutations. They can be caused by DNA damage. If you smoke, smoke contains carcinogens that will accelerate the mutations in your lung cells. Environmental carcinogens, plenty of those being spewed out by manufacturing. And infectious agents, including viruses, which is what we're going to talk about today. Transformation and oncogenesis are 2 different things. Transformation is changing the properties of cells and culture, all the things we've talked about, including them becoming immortal. But that's not enough to be cancer. You need more mutations, more changes in the cell than occur during transformation. Oncogenesis is the formation of a tumor that requires additional genetic changes. So this animal has a tumor on its back. That tumor started out as a collection of transformed cells that were immortal. And then here's what happens. When you let cells divide uncontrollably, that is very bad because they begin to accumulate mutations. And every DNA replication cycle, you accumulate mutations in your genome. Some of them are corrected, but some escape. And if you let cells divide uncontrollably, that is a recipe for formation of a tumor. So the cell gets transformed initially, keeps dividing, accumulating mutations, so you get those magic 12, and then you have a cancer. So cells that divide a lot, like our intestinal epithelial cells, intestinal tumors are quite common because of that. Now, what we understand about the relationship between transformation and oncogenesis, I would argue, is all from studying viruses. They sorted out the whole problem in the end. And what they showed us was that viruses can transform cells, and it gives them oncogenic potential. Transformed cells divide forever, they accumulate mutations, and they become cancers. No virus can do it all, which means viruses can transform cells, but the cancer arises because the cells keep dividing and accumulate mutations that eventually make them a cancer. Of course, the viruses started this, but the virus itself is not causing the cancer. That is a consequence of repeated genome replications, which is not supposed to happen. So here are viruses involved in human cancers. So they're called human cancer viruses, even though they're just triggering the road on the way to cancer. Epstein-Barr virus, these cancers, Burkitt's lymphoma, nasopharyngeal, and others we mentioned last time, hepatitis B and C virus, liver cancer, human T lymphotropic virus, a retrovirus, leukemia, HIV, many, many different cancers, because this is a persistent, as we'll see at the end of this course, it's a persistent infection. Your body is always making cytokines, and one of the things that cytokines does is to make cells divide. So you have cytokines around all the time, cells are always dividing, they get transformed, they get cancerous. Papillomavirus is a variety of cancers, Kaposi's sarcoma, herpes virus, a number of cancers, and Merkel cell polyomavirus, which causes a relatively rare skin cancer. So we think human viruses are the contributing factor in about 20% of human cancers. The rest is genetic and environmental. Now, the lesson for today is going to be Transformation and oncogenesis is not required for the replication of any virus. Any with an asterisk. So today I'm going to tell you this, what I want you to know is a story. This is one of the greatest stories in molecular biology. So let me tell it to you as a story that begins in 1909. This is something I've mentioned before. Peyton Rouse, he was working at the Rockefeller Medical Institute, which is what it was called at the time. Rumor has it that farmers would bring him chickens with big tumors on them. And here's one of these chickens here with a sarcoma, a solid tissue tumor. And he wanted to know what was causing this tumor. And it was 1909, not far after viruses had been discovered. And he showed that if you took this tumor from the chicken and ground it up and filtered it through 0.2 micron filters and took what passed through the filter and injected into a new chicken, that would cause a tumor. A cell-free filtrate. Remember the first definition of viruses, cell-free filtrate. He showed that cancer could be caused by a viral infection. Well, nobody believed him. 50 years later, though, he did get a Nobel Prize for this discovery. 50 years, the longest incubation period, as I've said before, for a Nobel Prize. He got it in 1966. His virus was called RSV, Rouse sarcoma virus. You can guess where the name came from. This turns out was worked on by many, many other people, and 2 more Nobel Prizes were given for work on Rouse sarcoma virus. So 3 Nobel Prizes from this chicken. A couple of years ago, a wonderful book was written. This is The Emperor of All Maladies. Siddhartha Mukherjee, who's a professor he's an oncologist at the medical center where I work, and he got a Pulitzer Prize for this. So here's a quote. By 1950s, cancer researchers had split into 3 feuding camps. The virologist, led by Rouse, claimed that viruses cause cancer, though no such virus had been found in human studies. Epidemiologists argued that chemicals caused cancer, although they could not offer a mechanistic explanation. The third camp possessed weak circumstantial evidence that genes internal to the cell might cause cancer. They were all right, and eventually it all comes together, as he puts it in this book. Now, Howard Temin plays a big role in this story. We've talked about him because he was one of the co-discoverers of reverse transcriptase. Remember that enzyme? Anyway, in 1951, a young guy went to Caltech. He was going to go study fruit flies, but he didn't like that. He decided to go work with Renato Dulbecco. Now, he was the person who developed the plaque assay for animal viruses. He was really very adventurous. When new things happened, he wanted to get on board. And one of the things Dulbecco was doing was growing cells in his laboratory, which very few people did in 1951. That was the year HeLa cells were isolated. Nobody could get anything but primary cells to grow well, and you didn't do a lot of experiments with them. But he was very adventurous, and he had cells growing in his lab, and Temin found out, and he came to use them. Now, you have to understand that up until this point, this virus could only be shown to be causing tumors in chickens. That's really hard to work on after a certain point. You can't figure out mechanisms. So Temin wanted to somehow make what he called cancer in a dish. Not really, but he wanted to study Rouse's effect in the dish. And eventually, after 7 years, he did that. What did he do? He took the virus and he added it to a layer of normal cells. Dulbecco was growing chicken cells in the laboratory. Easy to get. You take chick embryos out of eggs, take a fertilized chicken egg, crack it open, take the embryo out, chop it up, trypsinize it, put them in a dish, you have cells. He infected them with Rouse sarcoma virus. The infection incited them to grow uncontrollably. These are Mukherjee's words, forcing them to form tiny distorted heaps containing hundreds of cells that Temin called foci. The foci, Temin reasoned, represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably, pathological mitosis. All right, he's taking a little license here. This is not cancer. I've just told you it's transformed cells. Whether or not Mukherjee knows this, I'm not sure. It sounds really nice. He grew cancer in a petri dish, but it's not cancer. Nevertheless, what Temin did was to infect normal chicken cells, and you can see them. These are avian cells in the background, normal, and here are some foci of transformed cells, different kinds, low magnification, high magnification, spindly or round and refractile. So that's what he means. The virus transformed the cells, and this was the first time that anyone had seen an effect of this virus in anything other than a chicken. So Temin reasoned that he could study this and figure out what was going on. And he was absolutely right. So he started this entire field. Temin went on to discover reverse transcriptase, and he used Rous sarcoma virus. So he reasoned that the cells were permanently changed. You could pick out these foci of transformed cells, and they were immortal. They would grow forever. And at this point, we knew that Rous sarcoma virus had an RNA genome. And he said, there is no way that an RNA genome can permanently change a cell so that it's transformed. He said there must be a DNA intermediate. He was the one that thought of this. And so then he and Baltimore said there's got to be an enzyme that makes RNA into DNA. They both found it, and they both got the Nobel Prize at the same time for that discovery. So that's where the logic to find that enzyme came from. We also found that much later that DNA viruses can cause transformation of cells. In 1962, a polyoma virus was found to be able to transform baby hamster kidney cells. These are primary cells. Transform them, they keep growing. And in 1964, infection of mouse cells, Swiss 3T3 are mouse cells with a monkey virus, SV40, they grew out as transformed cells. And in these examples, the viruses killed most of these cells, but a few of them survived, and these were the transformed cells that lived forever. So we have early in the 20th century RNA tumor viruses, and then much later, DNA tumor viruses discovered. And the question we're going to address today is, how can a viral infection transform a cell? And there's three main requirements, which are shown here. First, we have to reduce or get rid of entirely cytopathic effects. The virus can't kill the cell, obviously. How could it be immortal if the virus killed it? Number two, we have to reduce virus replication, because that's often what kills a cell. And in general, these cells transformed by RNA and DNA tumor viruses do not produce virus particles. And finally, obviously, the cell has to continue to divide. If it's going to be immortal, it has to keep dividing. So these are three requirements for viral transformation of cells, and they should ring a bell, because it's exactly what's going on in persistence. Many of the persistent infections we talked about last time, no virus particles are produced. The cells are not killed. Same thing is going on here. So transformation is a type of persistent infection, and that's why I talk about it after we talk about persistent viral infection. Our first question is, which of the following is not a property of transformed cells? Increased requirements for growth factors, immortality, loss of anchorage dependence, loss of contact inhibition, colony formation in semi-solid media. So most of you got A, increased requirements for growth factor. That's not correct. There are decreased requirements for growth factors. All the others are characteristic of transformed cells. Now what I'd like to do is to go through how we figured out what RNA and DNA tumor viruses did in order to transform cells. And this is an amazing era in molecular biology, because in the end, it led to a unified theory of growth control of cells. It was viruses that showed the way. We have retroviruses discovered in the early 1900s. In vitro studies with Rous led to a convergence with in vitro cancer biology, so people are non-virologists trying to understand how cancer arose, and the DNA tumor viruses converged with this as well. And in the end, you'll see we understood how cell growth control was regulated by studies of these viruses. We'll start with the RNA tumor viruses and address the question, how does Rous sarcoma virus cause tumors? I should say, how does it transform cells and cause tumors in chickens? And again, that was started with the discovery of Peyton Rous in 1909. Now we need to know a little bit about chickens. And one fact is that all chicken flocks in the world, whether you raise them for meat or for eggs, they're infected with avian leukosis viruses, ALVs. They're endemic in all chicken flocks. These are retroviruses. And they were first discovered to cause these leukemias, leukosis, leukemia, a cancer of white blood cells, by Ellerman and Bang in 1908. So they actually discovered the first cancer virus, but as I said, in 1908, people didn't think leukemia was a cancer. So Rouse got the credit. Within a few months of hatching, most chickens get infected with ALV from their chicken mates, so it spreads throughout the colony. And in birds that are older than 14 weeks, about 3% of them develop leukemia spontaneously as a result of infection with this virus. We'll actually look at how this works in a moment, but first we're going to talk about Rouse's virus. The other 90% of the birds, they're infected, they have a transient viremia, their immune system clears the virus, and they don't get leukemia. However, as these birds get older, the ones that were infected, they develop other cancers. Some of them were sarcomas. These are connective tissue tumors, these solid tumors. That's the sarcoma type of tumor that Rouse worked on. If you grind up the tissue and inject it into a new chicken, they rapidly cause sarcomas, not leukemias. So the viruses, after these chickens recover and develop tumors, you can recover a different virus apparently from the tumor. But as you'll see, it's related to avian leukosis virus that causes a variety of tumors. And this is one of the viruses that Rouse isolated, Rouse sarcoma virus. And after he reported this, many other people repeated this experiment and got different kinds of Rouse sarcoma viruses. And most of them were defective. What that means is they're missing parts of their genome and they can't replicate without a helper Rouse sarcoma virus. Rouse was lucky because his isolate was not defective. There's nothing like luck, right? His virus was intact. Subsequent to his work, most of the other viruses identified from these kinds of studies in chicken were defective. After Temin's work showing that you could infect chicken cells in culture, a whole host of molecular biologists then said, how are these viruses transforming the cells? How does Rouse, but not avian leukosis virus, cause sarcomas? And a key finding was that the viruses, like Rouse sarcoma virus, had extra sequences in them. They were recombinants, and the extra sequence is a piece of host DNA. And what happens, as you will see in a moment, is that avian leukosis viruses, which cause leukemia, they somehow pick up a piece of cellular DNA, and that now makes them able to cause sarcomas. And Rouse sarcoma virus is one such virus. It's an avian leukosis virus that's picked up a segment of cell DNA. And these segments were called oncogenes because in the virus, they give rise to transformation. And you'll see these are cellular genes. And it's not a great name, but at the time when people named it, they didn't know what they were doing in the cell. As you'll see, these are genes that control proliferation. So they're not there to cause cancer, although if they are dysregulated, transformation will occur. These two individuals, Mike Bishop and Harold Varmus, figured out what was extra in Rouse sarcoma virus. They did this in 1976, and then in '89 they got the Nobel Prize for this discovery. And so that's the third Nobel Prize from Rouse sarcoma virus. The original Rouse, reverse transcriptase, and now the finding that Rouse sarcoma virus has picked up an oncogene from the host cell, and they called it SARC because the original tumor was a sarcoma, and it was abbreviated SRC. So there's Bishop and Varmus. They were working at UCSF at the time. I've interviewed both of them. TWIV episode 400, Harold Varmus, who was here at Sloan Kettering, he came up to Columbia and we recorded an interview where he told the whole story of how he was an English major in college and realized he wasn't very good at it and decided to go to medical school. And how he got into this whole idea of looking for the gene that Rouse sarcoma virus picked up. He, besides doing this and getting a Nobel Prize, he was head of NIH for 10 years and was responsible for doubling the NIH budget. He was then president of Sloan Kettering, and now he's just a professor at Sloan Kettering. I guess he's had enough. But they discovered that this virus picks up a segment of host DNA. They called it an oncogene. And this one picked up by Rouse sarcoma virus is called VSARC. So this was the major insight. So the idea here is that all birds come down with a variety of tumors. They start out being infected with avian leukosis virus. All the tumors that subsequently grow up in birds as they age are derived from ALV, but most of them were defective, and they're all different. I'll show you some of those in a moment. And again, Rouse was lucky because his Rouse was not defective. Each one of these viruses that were isolated by others had different host DNA, different from Rouse sarcoma virus. You took a chicken with a tumor that had naturally developed, pulled out the tumor and isolate the virus. It was a different cellular DNA piece that was picked up. Each segment had a novel chicken oncogene. This was a gold mine. Every month there was a new paper showing a new V something, VSARC, VMIB, etc. And this went on for quite a while. So here's a picture of some of these oncogenes. So we call these transducing retroviruses because they pick up a piece of DNA from the host cell, and when they infect new cells, they deliver it, and that causes the cell to be transformed. So on the left, we have our avian transducing retrovirus. Here's Rouse sarcoma virus. He isolated this from a solid tumor, a sarcoma, and what it is avian leukosis virus, that virus that infects every chicken in the world, including today, by the way. Avian leukosis virus picks up a piece of host DNA, and then it causes some kind of a tumor. So here's Rouse sarcoma virus. You can see an extra red sequence there called SARC. Avian leukosis virus, a typical retrovirus with a simple genome. This is the mRNA, GAG, Paul envelope genes. And you can see Rouse has GAG, Paul env, and it's got SARC extra at the end. So it's not defective. It's got the entire complement of retroviral genes. But all these others isolated from different kinds of tumors, myeloblastosis, myelocytomas, a different sarcoma, erythroblastosis, reticuloendotheliosis, different kinds of bird tumors. Again, you just let the birds get older, they develop cancers, and you isolate viruses from them. They're all different kinds of viruses. They have different oncogenes that they have picked up from the host cell, and they have different names. MIB, they're named after the disease pretty much. MIB, MIC, MIL, YES, ERB, REL. So it's a gold mine because each of these is a cellular gene. We call them oncogenes, but they're involved in growth control. You can do the same experiment with other animals, and people began to do that. Here are some mammalian transducing retroviruses. Same idea. You find a tumor in some animal, a mouse, and you isolate the virus. So here we have murine leukemia virus, which is the progenitor for many of these viruses. Here, Abelson leukemia virus, where Abel has replaced a good fraction of the viral genome. A sarcoma virus from mice. A cat sarcoma virus, another cat, a simian sarcoma virus, and the oncogene is cis, another cat virus, etc. So people did this over and over again with different species and identified different oncogenes that were picked up. And as you can see, most of these viruses are defective, except for Rouse's. How lucky was he? It's amazing. He didn't even know what he was doing, but he just happened to get a non-defective virus. If you're not understanding what I mean by defective and non-defective, a defective virus is missing a portion of the genome. And usually it's an envelope protein, but it can be more. The genes are deleted during the capture of the oncogene, as you will see in a moment. But here we have avian leukosis virus genome, again, GAG Paul envelope. Rouse sarcoma virus simply added SARC to the genome. It didn't delete anything, so that's non-defective. Whereas avian myeloblastosis virus, BA1, the MIB oncogene has inserted into envelope. Now envelope is gone. This virus cannot make particles. And so it needs a helper virus present in the same cell to provide the envelope in order for it to be packaged to a virus that could infect the new cell. That's what I mean by defective. It's missing a gene as a consequence of the insertion of the oncogene. Here's a model for how this oncogene capture happens. It's theoretical. There's some support for it, but I just want to show it to you because you may be thinking, how does this happen? How does the virus capture an oncogene? So here we have the viral double-stranded DNA made by reverse transcription. It goes in the nucleus and it integrates into host DNA. We've seen all this before, 2 LTRs, GAG-Pol envelope, and that is then transcribed by host polymerase to give you wild-type retroviral mRNA. Sometimes there is a deletion downstream of this construct, and these happen randomly. For example, here is a deletion removing the envelope gene. As a consequence, you also remove the right-hand LTR. Remember, that contains the transcription termination signal. So now, when the viral genome is transcribed by Pol2, the transcript goes past the Pol gene, and it includes some cellular sequences, which are shown in red here. Most of the time, this doesn't matter. No virus will be produced because this virus is defective. And if the gene downstream is not an oncogene, it won't matter. But sometimes, because these viruses integrate all over the chromosome, at some point it's going to sit down next to an oncogene. And then it can capture it. So some of these red sequences will be oncogenes. They'll be packaged into virus particles. And even if there's only one copy of the defective genome with the oncogene, again in red, copy choice, remember, doing reverse transcription can generate a particle with the oncogene in it. And eventually that will infect the cell. It will be reverse transcribed. The oncogene will be produced and the cell will become transformed. And what we have learned is that not only do viruses pick up oncogenes in this way, but as they grow, they sustain mutations and rearrangements of the oncogenes that make them more potent. So that's how we think, or at least one way, this is one mechanism for capturing an oncogene by a retrovirus, but it is accidental. It actually has nothing to do with the retrovirus life cycle, except that the retrovirus must integrate its DNA, but capturing an oncogene is not part of the productive infectious cycle of a retrovirus. Now, so far we have identified over 60 different cellular genes that can be picked up by RNA tumor viruses to cause transformation. And all of these genes, they're called, in the cell genome, they're called proto-oncogenes because they're not normally involved in transforming cells. They're all involved in the control of cell growth. They're involved in the regulation of the cell cycle. We'll look into that in a moment. They're all highly regulated because you don't want them on all the time. You don't want cells dividing all the time. So the proto-oncogenes, the normal cellular genes, have these kinds of abbreviations like C-SARC, C-MYC, MOS, RAS, etc. for all of those genes picked up by the transforming viruses. And then the virus copies are called V, SARC, V-MYC, etc. Sometimes these are unaltered copies of the cellular gene, but as I said, often they contain mutations and rearrangements that even increase their transforming ability. So these are proto-oncogenes. And if you look at all of these genes and what they encode, that's shown here. They encode the entire signaling pathway that regulates cell division, the control of mitosis. Remember, the original phenotype is transformation, uncontrolled cell growth. And so it makes sense that these genes that are picked up by retroviruses that cause uncontrolled cell growth are involved in its control. And these genes encode proteins that can be growth factors. So one of the oncogenes picked up by a retrovirus called CIS is an actual soluble growth factor. And of course, what a growth factor does, you add it to a cell, it stimulates the cell to divide. That's what's in serum. When you grow cells in serum, they have lots of growth factors in them, and they bind to receptors on the cell surface, and they make them divide. That's how you do cell culture. So some of the oncogenes are growth factors. Some of them are receptors for growth factors. And then some of those are components of the signaling pathway that leads to mitosis, like membrane-bound protein kinases, G proteins, GTP exchange proteins, cytoplasmic protein kinases. Again, these are all oncogenes. And transcriptional regulators in the nucleus, transcription factors that bind to genes that induce cell division. Every one of these, if picked up by a retrovirus, It is then inappropriately expressed. There's no regulation of its synthesis, and that causes the cell to divide. And you could look at every stage here. Yeah, if I put a lot of growth factors in the cell, they're going to divide. If I put these GTP exchange proteins in, they're going to divide. If I put transcription factors in that control cell growth, they're going to divide. That's how these RNA tumor viruses work. They make the cell grow by inappropriately expressing these, and one at a time. Only one at a time is needed to cause cells to grow uncontrollably. And again, this we figured out from our study of RNA tumor viruses. So here we have to look at the cell cycle to put this in perspective. And here it is, of course, the single cell cycle beginning at mitosis and then followed by gap phase 1. There's another phase of synthesis, DNA synthesis, of course, and then we go through cell division. So that's the basic cell cycle. These proto-oncogenes that we've been talking about that are picked up by RNA tumor viruses, these act right up here at G0. They push the cells into division, and they send them around the cell cycle. So these are mitogenic signals encoded by each of those oncogenes. We call them dominant oncogenes because you just need one copy to put in a cell and it will push it into cell division. And they were all revealed by studying these retroviruses. So today's understanding of what controls cell division is, as I said before, largely obtained by studying these viruses. The way these viruses transform is not all the same. There are 3 distinct mechanisms. What I've been telling you so far is summarized in the first 2. We have first rapid tumor formation. You take Rous sarcoma virus, you inject it into a chicken. Within 2 weeks, you have a tumor. The virus infects the cells. It produces VSarc. VSarc starts the cells dividing, and then you have the tumor produced. Next, you have viruses with intermediate kinetics, and this is like the avian leukosis viruses. It takes months for tumors to develop. There's no oncogene in ALV. Remember, ALV is the parent of Rous sarcoma virus. ALV doesn't have an oncogene. So how does it cause leukemias? Well, it turns on expression of endogenous oncogenes. We'll see a picture of it, but picture the provirus integrating. It's got a LTR on the right side with a promoter. Promoter can activate an oncogene. That's how these viruses work with intermediate kinetics of tumor formation. And finally, we discovered not too long ago, human T-cell leukemia virus, HTLV, takes years to induce a tumor. These are human tumor-causing viruses. They have no oncogene. They do not cause cis activation of local oncogenes, as the first 2 viruses do. But what they do is they make regulatory proteins, transcriptional activators that they require for their genome, and it turns out that they hit some cellular genes and turn on their synthesis. And if one of those is an oncogene, then you have transformation. That is all illustrated here. 3 different mechanisms of transforming cells. We have rapid induction of transformation, like Rous sarcoma virus. The virus is a transducing retrovirus. It carries an oncogene, which is picked up in a previous replication cycle. So 2 LTRs here, integrates into the host cell genome, and that oncogene is then expressed as part of the production of messenger RNA. The virus makes a lot of protein and it kicks the cells into dividing, and you get rapid tumor formation. Cis-activating viruses like avian leukosis virus, they don't carry oncogenes in their genome. You see GAG-Pol envelope, nothing else. What they do is they integrate randomly, and occasionally they will integrate next to one of those 60 oncogenes that is involved in growth control of the cell, and the right LTR has a promoter in it. The 2 LTRs are identical. They both have promoters and terminators. So the promoter in the right LTR will work, transcribe the oncogene that it's integrated next to, and that causes transformation. And then we have the slow transformation viruses that are trans-activating, no oncogene, no cis activation of an oncogene, but rather, in this particular case, they encode a transcriptional activator, a protein that's needed to transcribe viral genes. And this happens to activate certain cellular genes, and if one of those is an oncogene, you will have long-term transformation. One of these that's activated by HTLV happens to be IL-2, a protein that can cause T cells to proliferate. And that's how this causes T cell leukemias. So mammalian transforming retroviruses transform cells is a mistake. This is not required for viral replication. It happens because their DNA has to integrate. Retroviral DNA has to integrate into your DNA in order for the virus to be transcribed. And that step accidentally results in capture of oncogene or cis activation. There is no requirement for transformation or oncogenesis. In order for a retrovirus to replicate, the cell doesn't have to be transformed, it doesn't have to be a tumor. I want to emphasize that this is completely an accident of viral replication. There is actually, though, one virus that we know of where tumor formation is actually needed for the virus to spread, and that is the walleye dermal sarcoma virus. That's a walleye, in case you didn't know. It's a fish that people like to eat, but they develop tumors. And the tumors, there's one. It's caused by a sarcoma virus, very much like the sarcoma viruses I've told you, except they're specific for fish. And these tumors develop on the fish. They're fine. And in the fall, the tumors fall off. They're full of virus. They go into the water, and other fish acquire the infection. And then the cycle is repeated. So the tumor is needed to transmit the virus to other fish. So in this case, the transformation is needed for the virus to propagate. Very interesting mechanism. But as far as I know, there are no other examples of this. And certainly the mammalian tumor viruses do not require transformation in order for them to propagate. Next question is, which of the following allows Rous sarcoma virus to transform cells? Presence of the envelope gene, presence of a Pol gene, presence of a Sarc gene, presence of LTRs, none of the above. Most of you got presence of a Sarc gene, which is right. Certainly not presence of an envelope gene. Because remember, many of those transforming retroviruses have lost the envelope gene. But Rous has it, but that is not what really transforms the cells. It's the Sarc. And none of the above wouldn't be right, of course, because it certainly is Sarc. The cellular gene picked up by that particular retrovirus. All right, let's turn to DNA tumor viruses and see how they transform cells. Remember, we learned that DNA viruses can transform cells just like RNA viruses, tumor viruses can. Let's see how that was sorted out and how it converges with what we learned from the study of retroviruses. So have any of you heard of a jackalope? A supposedly mythical creature seen in the wild is nothing more than a rabbit with papillomas on its head. And people from a distance think they look like a hybrid between a rabbit, a jackrabbit, and an antelope, so they call them jackalopes. You can Google it, you'll find it. They don't exist, they're just rabbits. And there are two examples. These guys have extensive papillomas growing from their faces. These are warts, and they fall off. The rabbits are okay. They don't look very happy, do they? This guy looks very angry. But they don't harm the rabbits. But in 1933, Richard Shope, a virologist, found that these were caused by a virus. These are cottontail rabbits, and it's a papillomavirus. He called it a papillomavirus. He was the first one, I believe, and a papilloma is a wart. And so now we know there are many different types of papillomaviruses, but this is a type of transformation of cells. These are benign tumors. They don't kill the animals, but they are certainly transformation of cells. Subsequent to that, it was found that a murine polyomavirus could cause tumors under certain conditions. So these viruses are mouse viruses. In mice, they don't cause any cancers. But if you infect hamsters and rats and rabbits, they make tumors in different tissues. And that's why they were called by Ludwig Gross polyomaviruses, because polyoma, many tumors. So a polyomavirus is a small DNA-containing virus. SV40 is a polyomavirus. Papillomaviruses are very similar as well. So 1953, another example of a DNA tumor virus. And finally, in 1962, Eddy and Hilleman showed that SV40 could cause tumors in newborn hamsters. SV40 is a monkey polyomavirus. The reason they discovered SV40 is because it was a contaminant of early polio vaccines. These vaccines were grown in monkey kidney cell culture, primary monkey kidney cells. They would capture monkeys, take out the kidneys, and make cell cultures and use them to grow the polio vaccine. And they didn't know anything about SV40, so it turned out it was a contaminant. It turned out many million Americans got infected by immunization, both with the inactivated vaccine and then subsequently with the oral infectious vaccine. But as far as we know, it didn't cause any tumors in any humans, although many lawsuits were brought up saying that these individuals' tumors were caused by SV40. There was never any proof of that. This virus infects monkeys, doesn't cause any tumors in monkeys, and it doesn't transform monkey kidney cells or monkey cells in culture. So again, just like papillomaviruses, the tumors are being formed in the wrong host. It's not the natural host of the virus. And that's going to be important for understanding how these work. Here's a summary of these data with DNA tumor viruses. We have this animal species that's being infected here on the left, and we have the virus SV40 or mouse polyomavirus, these two DNA tumor viruses. So in the monkey, SV40 is permissive. It replicates, kills monkey cells, no tumors. It's the natural host of the virus. SV40 in monkeys is permissive. Mouse polyomavirus, if you infect monkeys, the cells are non-permissive, the virus doesn't get anywhere, no tumors. The mouse, you infect with SV40, the cells are non-permissive, so no tumors. Mouse polyomavirus, the mouse is the natural host, the cells are completely permissive and they die. Then we get to hamsters. Hamsters are semi-permissive for both SV40 and mouse polyomavirus. We see tumors in hamsters infected with either of these viruses. In rats, these cells are semi-permissive for both SV40 and mouse polyomavirus, and we see tumors there. What do I mean by semi-permissive? No late genes are expressed. Remember, these DNA viruses have transcriptional programs. They have an early and a late phase, sometimes multiple earlies, but they all have a late phase. Semi-permissive means there's no late phase of replication. Another important fact is that the transformation of cells in culture by these viruses is pretty rare, about 1 in 100,000 infected cells. And so we want to figure out why is it rare and how does this transformation relate to tumor formation in the animal. One more virus that we have to put on our radar, and that is adenovirus, a different family. Remember, long, double-stranded linear DNA molecules with the Sputnik-like virus particles, lots of human serotypes. None of these cause cancers in humans. But if you infect hamsters with different adenovirus serotypes, you will get some tumor formation. And just like tumor formation by the polyomaviruses and the papillomaviruses, tumor formation by adenoviruses in the wrong host is very rare. All right, so now we're getting some similarities among these different viruses. Then people started to look in cells transformed by SV40, papilloma, and adenovirus, and they found one thing in common. They all produce T antigens. And in many cases, nothing else. This is why I told you about T antigens earlier in the course, so you would now understand them. For SV40, large T antigen. In the tumors in hamsters caused by SV40, they only found large T antigen present. And just to remind you, that's large T up at the top there, with all its different functional domains. The polyomaviruses also encode T antigens. Papillomaviruses, they're encoded by these genes, E5, E6, E7. In adenoviruses, the E1A and the E1B genes encode those proteins that are also T antigens. And they were called T antigens originally because they were found in tumors way before people learned of their role in DNA replication and transcription and so forth. So these are all found in transformed cells transformed by these viruses. They all have these T antigens. And these T antigens are all different. SV40T is very different from polyoma, papilloma, and adenovirus T antigen. All right, so that's an observation. What does it mean? Remember that T antigens are essential genes of these viruses. They're required for replication. They activate transcription. We need them for DNA synthesis. They bind the origin for SV40, for example, and melt it and attract the DNA synthetic apparatus. And again, they are the only genes, the only viral genes that we find in these transformed cells, transformed by these three different kinds of viruses. And now we know that you can take a gene encoding a T antigen on a plasmid by itself, and you can put that into a primary cell and you can immortalize it. It's a common way to make cell lines. So you can take your cheek cells and get a plasmid with SV40 T antigen on it, put it into those cells and make an immortal cell line which will outlive you. So if you so wish to have your cells outlive you, you can do that. Just make sure you freeze them, because no one will split them once you're gone. T antigen alone can transform cultured cells. And these three discoveries here tied all of this together, made in many different laboratories. First, SV40 T antigen was found to bind a cell protein of molecular weight 53,000 Daltons. This protein came to be known as P53, which is an amazingly famous protein. It's mutated in almost every human cancer, and I think you'll understand why in a moment. Second, and this is something we've talked about before, early adenovirus genes, their transcription, the E2 genes, requires a cell transcription factor called E2F. We've talked about this before. And finally, E2F, this cellular transcription protein that's needed for the transcription of the adenovirus early genes, the E2 genes, binds to a cell protein called RB, retinoblastoma protein. And all of these proteins, P53, RB, and E2F, all will come together in a few moments and explain how the cell cycle growth is controlled and how these viruses disrupt it. Now here we go back to our cell cycle. We showed this cycle at the bottom before. Mitosis is expanded here just so you can see how the cells divide, but it's actually not crucial for our discussion. We know from the work with RNA tumor viruses that those oncogenes function to kick the cells into mitosis. But there is another restriction point in G1, right down here. So even if you get cells to go through mitosis, they won't proceed until they get past that restriction point. And that's what we're going to explore right now. And of course, if they don't go through the restriction point, they're not going to divide anymore because they won't make DNA, and they will never get to that next mitotic cycle. This second restriction point, shown by the red line, that's governed by cell proteins which ask, are conditions right in order to make DNA? Are there enough nutrients around? Are other conditions right? And you'll see what some of those conditions are. So even if the proto-oncogenes, the mitogenic signals discovered by RNA tumor viruses, even if they get the cell through mitosis, it's going to stop here unless the external environment, and as you'll see, unless the nuclear environment is the right one as well. So what am I talking about here? This restriction point is actually governed by RB, the retinoblastoma protein that was found to bind the adenovirus T antigen. It was first discovered in children who have retinal tumors. The tumors are of the cells called retinoblasts. These form the retina. They're gone by age 5. So these are tumors you only see in kids who are less than 5 years of age. They develop these tumors because they have lost both copies of the gene encoding the retinoblastoma protein. So this is a recessive oncogene. If you have the wild-type protein present, it acts at this restriction point and prevents cells from dividing uncontrollably. If both copies are missing, there's no longer a restriction here. The cells can keep dividing, and eventually, as you know, that leads to transformation and eventually cancer. So this restriction point, that is mediated by RB. And that binds the T antigens of adenoviruses. So here's how this whole thing works. On the left is the mitogenic control pathway, which was discovered by the study of RNA tumor viruses. We have on the surface receptors that bind growth factors. And all of these components are oncogenes. The growth factors bind a receptor. There is a signaling pathway that is initiated. And these proteins, SHC, GRB, RAS, they're also named because they were discovered in RNA tumor viruses. They eventually initiate a signaling cascade that turns on mitosis and phosphorylates RB. That's needed to get to the G1S transition. So not only do you initiate cell division through these various pathways that are shown here, the cyclins have to be increased, this protein has to be degraded, P27, KIP1, but you also have to phosphorylate RB. And that's because RB is normally complex to the E2F transcription proteins. Remember, E2F, family of transcription proteins needed for adenovirus transcription, but those are also needed to transcribe the genes that are needed to go through mitosis. So as long as RB and E2F are bound up, this recruits a histone deacetylase to the promoters that are recognized by the E2F proteins. It inhibits transcription of genes needed for DNA synthesis and mitosis. If you phosphorylate RB, which happens when you have signaling from the cell surface, RB and its histone deacetylase comes off of E2F. E2F can now bind to promoters that turn on genes involved in DNA synthesis and mitosis. So that's how RB is a checkpoint regulator, because if it is unphosphorylated, it will bind up E2F and prevent the synthesis of the genes that are needed for mitosis. So that couples together the mitogenic pathway and the checkpoint regulation all together there. Remember, though, that DNA viruses do not replicate in quiescent cells. So a cell in which RB is bound up to E2F is not dividing, and DNA viruses cannot replicate in them because the DNA synthetic machinery is not accessible to them. So they have to somehow get past that. And we talked several lectures ago about how T antigens push the cells into cell division. They push them past this checkpoint. How they do so is they bind RB. Here's RB complexed again with E2F, silencing the genes that are needed for DNA synthesis and mitosis. The T antigens, shown as these purple blobs, large T, papillomavirus E7, adenovirus E1A, they all bind RB, and they pull the histone deacetylase away from promoters so they can be active. Those promoters control DNA synthesis and mitosis. So now we see the connection between T antigens and RB. They bind it, move it away from the promoters that are needed for mitosis. Another step that is regulated here is mediated by P53. So remember, T antigens, in particular SV40 T antigen, was very early on found to bind this protein called P53. And we now know that P53, one of its functions, is to monitor DNA damage or unscheduled DNA synthesis. In other words, if the cell is not supposed to be dividing and it finds that there is some replicating DNA in the cell, it will kill the cell. So here's an example of that. We have a double-stranded DNA break. The P53 recognizes that. There's a whole sensing system, which you don't need to understand here, that results in modification of P53. It then tetramerizes and activates P53 responsive promoters of genes that cause cell cycle arrest and cell death. So if P53 senses DNA breaks or unscheduled DNA synthesis like that, which would occur in a virus-infected cell, P53 initiates a cell-killing cascade. The reason is that the P53 is there so that we don't duplicate damaged DNA if it's not been repaired by our repair proteins. Now, obviously, viruses have to counter this because as soon as DNA viruses infect a cell, they will do unscheduled DNA synthesis. They'll kick the cell into dividing as well as they'll be dividing their own genomes, and that freaks out P53, and P53 is going to kill those cells. So DNA viruses have to counter it. And they do in many ways, shown here. Here's P53, and these 1, 2, 3, 4 different ways of viral proteins countering the activity of P53. So P53 accumulation, again, would lead to cell cycle arrest and programmed cell death, apoptosis. Here we have a adenovirus E1b protein, which is binding to P53 and preventing it from inducing these genes. Here we have SV40 large T. Many copies of it sequester P53, so it can't act to turn on the cell-killing pathways. The papillomavirus T antigens in red and orange, they direct P53 to be degraded by the proteasome. And so does adenovirus proteins. Adenovirus E4, ORF6, and E1b, they bind P53, and they have it also degraded by the proteasome. So each of these DNA viruses in some way negates the activity of P53, so it doesn't send the cells into death. Obviously, the viruses need to do that, otherwise the cell would die and they wouldn't be able to replicate, right? Makes perfect sense. So every DNA virus that kicks cells into dividing so that they can replicate their genomes, most of them do, not the pox viruses, they all encode inhibitors of P53-mediated cell death. Our last question is, what are T antigens? They're encoded by viral genes that are essential for replication. They're present in tumors and transformed cells. They're encoded by viral genes that have been incorporated into the cell genome. They're antagonists of cell cycle checkpoint proteins, or all of the above. All right, all of the above, that's right. These are all features of T antigens. Two more things we have to figure out. First of all, why is only T antigen produced in these transformed cells, and why is transformation so rare? It's rare because two low probability events have to occur. First of all, you cannot produce late genes. These are typically lethal. They give rise to virus particles, and that can end up killing cells. So you either have to have a rare deletion of late genes to transform a cell, or you infect a semi-permissive cell where there's no late gene expression. That's why a monkey virus infecting a hamster, where it's non-permissive, can lead to transformation. And secondly, T antigen has to be on all the time, and it's got to be transmitted to every new cell that's produced, which means it has to be integrated into the host cell DNA. So these are two rare events: getting rid of late gene production, integration into the genome. That's why transformation is so rare when we do these infections of the wrong cells with various viruses. So transformation, tumor formation, these are abnormal events. They are a result of the fact that these viruses have to kick the cells into DNA synthesis. They are not required for normal replication. Remember, in a permissive host, we never see transformation. And they were discovered by infecting the wrong hosts: a human adenovirus in a hamster, a simian polyomavirus in a mouse. Now, caveat, we now know that even in the natural host, sometimes those two rare events occur. Late gene expression doesn't happen, integration of T antigen occurs, and we now know that human papillomaviruses, which can cause cancers in humans, it's the right host, yet they still can cause transformation as tumor eventually. It's what happened to Henrietta Lacks. Apparently, these rare events can also happen in a natural host, but we discovered them in the wrong host. So transformation, I like to say, is an epiphenomena of two unique reproductive cycles. Epiphenomena is something that arises from but doesn't influence a process. DNA tumor viruses have to get the cells dividing. T antigens do that. They get the cells dividing and they inactivate this checkpoint protein. They also inactivate P53. But these are normal functions of this T antigens that aid in replication. But if they're constitutively produced and the cell doesn't die because you've gotten rid of late genes, then they lead to transformation. And remember, they're transformed and may be on their way to becoming cancer cells. So this summarizes both sets of viruses, emphasizes for both classes, again, transformation are epiphenomena because RNA tumor viruses have to integrate. So the chances that they will pick up by accident an oncogene, and DNA tumor viruses have to turn on the cell cycle and antagonize RB and P53. So we've discovered proto-oncogenes, genes that regulate the mitogenic cycle, that get the cell cycle through mitosis. These are all what we call dominant oncogenes. And then the DNA tumor viruses revealed how the checkpoints work. And these we call recessive oncogenes. And what I really find amazing is it all began with a chicken with a tumor and curiosity.